Yes. Hello students. In yesterday's class we were discussing about the nature of bonding. See, when you take the complex food molecules, that is complex macromolecules like protein, polysaccharide, nucleic acids, and one more is lipid. See, these all complex food molecules are made by smaller units, that is we can call it as monomeric units. Like protein is made up of monomeric units of amino acids, polysaccharide monosaccharides, lipid fatty acids or glycerides, and nucleic acid polysaccharide. So what kind of bond is present in between those? That is, when you take protein, peptide bond. When you take polysaccharide, 1,4-glycosidic bond or alpha 1,4-glycosidic bond, and one more is when you take lipid, ester bond, and when you take nucleic acid, there are three bonds. One is the bond present in between pentose sugar and nitrogenous bases, that is glycosidic bond. The bond present between nucleoside and phosphate, ester bond, and the bond present in between the two nucleotides is called as phosphodiester bonding. And continue the same topic, that is we are in detail moving on to the nucleic acid, that is DNA and RNA, and more specifically I am talking about the DNA. So the structure of DNA is the better example, as we discussed in the earlier classes, that is primary structure of protein, secondary, tertiary, and quaternary. Likewise, the DNA has a structure which is secondary structure, and the DNA contains some of the ingredients or the chemical composition, that is pentose sugar, also called as ribose sugar, which contain 5 carbon atoms. Second one is nitrogenous bases. There are two types of nitrogenous bases. There is purines and pyrimidines. The purines are of adenine guanine, pyrimidines are of cytosine and thymine. These two are double ringed and these two are single ringed, heterocyclic. So, and last one is most important, the chemical composition is phosphoric acid is in the form of phosphate. Yes, and by taking all these ingredients we take the structure of DNA. See, yes, this is the second structure of the DNA, and we'll write pointwise. First point, see, the DNA is first point is DNA is double stranded. See, DNA is a double stranded. See, this is first strand and this is second strand. This is first point. DNA is always double stranded. In that, one strand, important word, one strand is in the direction of 3' to 5'. That is 3' to the same end ends with the 5' or 3' prime, and another strand or second strand is in the direction of 5' to 3'. That is like this, 5', same end ends with the 3'. Is that clear? Second point. See, these two strands run in opposite direction. I mean to say, one strand is in the direction of 3' to 5', and opposite strand is run in the direction of 5' to 3'. Hence, these two are called as anti-parallel. They are nothing but anti-parallel. So the word anti-parallel refers to one will be the direction of 3' to 5', another one will be 5' to 3'. Is that clear? This is one. Next, in this DNA strand, there are 4 nucleotide bases. there are 4 nucleotide bases. So those 4 nucleotide bases are, one will be adenine, we can call it as adenylic acid, guanylic acid, cytidilic acid and thymidilic acid. Also called as deoxyadenylic acid, deoxyguanylic acid, deoxycytidilic acid, deoxythymidilic acid. So these 4 different nucleotide bases are present in the DNA. And the most important thing to understand is where these 4 deoxy, that is nucleotide bases, are present in the structure, where they are present. See, these are, in one strand, if there is a adenylic acid, opposite strand will be having thymine. Very important sentence. So in that, A or adenylic acid is present in one strand, that is in the 3' end. Same time, the thymidilic acid or deoxythymidilic acid is present in the opposite strand, that is in the 5' end. So I mean to say that if adenylic acid is present in one strand, definitely in opposite strand we must have, that is thymidilic acid or deoxythymidilic acid, so that these two are called as complementary to each other. Simple word is, these two are called as complementary to each other. The word complementary to each other means, when I write A, we must remember there must be a thymine. It's understood. Hence these two are considered as complementary to each other. If I write thymine, you must remember there must be a adenine. Hence it is called as, I mean to say that, better word I can tell you people, if A is present in one strand, I can predict T must be present in the opposite strand. Hence it is called as complementary strand. Same time, if I write cytosine or cytidilic acid, there must be a deoxyguanylic acid. I mean to say, if C is present in one strand, G is present in the other strand. This is called as complementary strands. Hence these two strands are called as complementary strands. These two strands are complementary to each other. Next important sentence is, if adenylic acid or adenine is present in one strand, as I told you, thymine is present in the opposite strand, hence called as complementary to each other. But very important thing is that, so this A and T are held together by 2 hydrogen bonds or double bonds. See, A, T are held together by double bonds. Is that clear? Yes. Next, when I write cytidilic acid and guanylic acid, like this, so again this one, and here these 3 are held together by 3 hydrogen bonds. We can call it as triple hydrogen bonds. Clear? This is another thing. See, if I mean to say that A always pairs with a T, or adenine always pairs with a thymine with 2 hydrogen bond or double bonds, and cytosine always pairs with the guanine with a triple hydrogen bonds. This is one important feature in case of DNA. Next word is backbone. So when you see the structure of DNA, the backbone is nothing but, backbone means, so where I draw this one, the main structure or thread like structure is called as backbone. So that backbone is made up of pentose sugar. CET question, remember. I mean to say, in the DNA structure, who is the backbone? Yes or no? So I am standing in front of you people. How? Because of presence of a backbone. It's called as vertebral column. And same time here, the who is representing? Who is representing the backbone? That is pentose sugar. Clear this? This is one. Next, so when you take this here, I mean to say, when you look at this structure like this, This is, I mean to say, from here to here. This is called as one groove. This is called as one groove. Do remember? I mean, it is like a staircase. Yes, it is like a staircase. So, yes, shaped one staircase. So, from here to, I mean to say like this. This is from here to here. It is considered as one groove. That one groove is always, I mean, the measures about 34 angstrom. It measures about almost 34 angstrom. In that, each groove again contain 10 base pairs. PP refers to base pairs. Remember, see how CG, GC like this, and AT, TA, again AT, CG like this. So there are 10 base pairs present in each group, right from here. Again AT, GC like this, GC whatever. From here to here, there are 10 nucleotide base pairs, not base. A is called as base. AT together called as one base pair. Likewise, in each group, I mean from here to here, there must be 10 base pairs. Is that clear? 10 base pairs. And now we can calculate. Each groove measures about 34 angstrom. I told you people, right from here to here it is 34 angstrom. Yes. And each groove contain 10 base pairs. Each groove contain 10 base pairs. Now the question is that what is the distance between each base pair? Each base pair. Yes, what is the distance between each base pair? I mean to say that, imagine I am asking this distance. That is 34 divided by 10. That is each base pair measures about, I mean distance between each base pair is 3.4 angstrom. 3.4 angstrom. So that is what? The distance between each nucleotide. So that CET question or NEET question, remember, how can we calculate the length of the DNA? Length of the DNA. See, very simple. By understanding this concept, we can tell whatever the length of the DNA. Say, here we have a length of DNA. Is what is the length of this DNA? First we need to understand how many base pairs are present. How many base pairs are present in this? So say for example there are 100 base pairs. There are 100 base pairs. Now tell me what is the length of the DNA? Simple to calculate. Number of base pairs into distance between each nucleotide bases, that is 0.34 nanometer or 3.4 angstrom. Both are one and the same. So here the simple calculation is 100 into 0.34 nanometer. That can give you the length of the DNA. Very simple. So in case of human beings, so there are 6.6 into 10 raised to 9 base pairs are there. In human, I mean in mammalian cell, or in case of human body, one single cell nucleus contain 6.6 into 10 raised to 9 base pairs. Then what is the length? 6.6 into 10 raised to 9 into 0.34 nanometer. That can give you almost 2.24 meter of DNA. Yes. So the thing is that in human body, a single cell contain almost 2.24 meter of DNA. How I calculated this one? Simple. Number of base pairs in the DNA into the distance between two successive nucleotide bases. That can give you the length of the DNA. And again, still many things to understand. Yes. In that, a single base pair. Yes. So a single, I mean two base pairs. See for example, take these two base pair. It can turn. That is, each base pair can turn. Yes. How much? 36 degree. Each turn can turn 36 degree. Now simply you can calculate each groove can turn into how much? That is, I told you people, single, that is this one, from this one to this one, it can turn 36 degree, means there are 10 base pairs, so 36 into 10 is equal to 360 degree. So simple, so that into 10, because 10 base pairs are present in each groove, and one base pair can turn 10 degree, sorry 36 degree. 36 degree means one base pair, and there are 10 base pairs present in each groove, so that there are, one single groove can turn 360 degree. Do you remember this point? Yes, this is what one thing. And we have almost, that is, when you take DNA, their diameter is almost 20 angstrom. 20 angstrom or 2.0 nanometer, both are one and the same. This is the diameter here. 20 angstrom or 2.0 nanometer. This is the diameter of the DNA. So this is also one of the important point to consider. And here, so again, in the long length of DNA, do you remember Charge-Graph's rule? See, write down Charge-Graph's rule. Here the word Charge-Graph's rule means total amount of purines is always equals to pyrimidines. Clear? Very simple. So total amount of purines present in the DNA is always equals to pyrimidines present in the same DNA. Nothing but, see, purines are nothing but A plus G, that is adenine and guanine are nothing but adenines. So total amount of adenine and guanine is always equals to cytosine plus thymine. Yes or no? This is what? Nothing but A plus T divided by G plus C is always equals to 1 or always constant. This is what? Charge-Graph rule. Simple. Total amount of purines is always equals to amount of pyrimidines. This is also one of the point included in the nucleic acid. So, and here, any other things to discuss? So these two are anti-parallel. A always pairs with the T and T always pairs with the A. G always with the C, C with the G, with the double and triple bonds respectively, and each groove, that is almost 34 angstrom. Each groove contains 10 base pairs. Yes or no? So this is one thing, and it is double stranded. These two are anti-parallel, etc. This is our DNA, how it is present. And most important word is this DNA acts as genetic material. This DNA acts as a genetic material. So the word genetic material refers to which can carry the information from one generation to the next generation. So that I mean to say that in the form of gene, as we know that in the form of gene. See, when you take the long length of DNA, in that somewhere you will be having a functional segment, somewhere you don't know where it is present, but in this long length of DNA, somewhere you will be having the functional segment of this DNA. That functional segment of DNA carries at least some kind of information. So it may be trait, or it may be character, or it may be protein synthesis, whatever, enzyme synthesis, whatever. So that is considered as gene. That nothing but which carry the functional information or some genetic information. That is nothing but gene. Yes. So this is about the structure of nucleic acids. Hope you understand this concept. And so till now we discussed about the protein. Yes, right from beginning. We conducted one experiment, that is chemical analysis. Second one is destructive experiment. By these two experiments, we separated various organic, inorganic elements, compounds, and one more is the molecules. This is what. and later how the DNA is formed, how the nucleic acids are formed, how the proteins are arranged, how the proteins are, and they're made up of what? Lipids, polysaccharides, macromolecules, micromolecules. These all we discussed in the in earlier classes. And in next class we are moving on to the very important topic, that is dynamic state of body, that is dynamic state of body constituents, nothing but concept of metabolism, is concept of metabolism. Dynamic state of body constituents, nothing but concept of metabolism. In that I can tell today some important concepts. As you can understand, dynamic state of body constituents, dynamic state of, so the word dynamic state, first word is dynamic state. What is dynamic state? Simple thing to understand. Dynamic state refers to, dynamic state of body constituents. The word constituents means materials. It may be elements, it may be complex food molecule, it may be simple one, it may be macromolecule, it may be a simple, that is micromolecule, whatever, irrespective. In that, those all constituents are in dynamic state. Dynamic state means they are always in a functional nature. They are performing some other functions, whatever the function, but they are performing the function. So nothing but dynamic. Simple? Yes. So dynamic state of body constituents, in that concept of metabolism. I mean to say that when you go to hospital, when you check your blood glucose level, blood calcium level, creatinine, lipid level, fatty acids level, ketone bodies, yes or no, sodium level, then magnesium level, iron level, yes or no. So that what I am going to impress on you is that in our body, so some of the very important, or they are already made some standards, yes or no. Say for example, a healthy individual, healthy male, so must contain almost 12 to 16 or 17 the hemoglobin. It is standard, that is 16 to, imagine 16 to or 12 to 16 gram percent, that is standard hemoglobin percent. Yes or no? At the same time BP must be 120 by 80. 120 by 80. So likewise, in our body, each and every constituent must have the standard level in the body. They must be present, and those levels and those standard levels should not cross the beyond limit or should not come below the normal level. Yes or no? So that it is normal. Imagine the hemoglobin, the adult normal hemoglobin level is, that is in case of adult human body, the normal hemoglobin level must be 12 to 16 gram percent, imagine. So that it should not cross 16, no doubt nothing will happen, but it should not cross, and it should not come below 12 also. Sometime it may be 6, 7 or 8. So nothing but they are suffering from anemia or some other different problems. Leave that. I mean to say that they should not cross the beyond limit or should not come below the normal level. Likewise, when you take any cell, it may be bacteria, it may be protozoa, it may be a single mammalian cell, whatever the cell you take, so those all cells are made up of, whatever the cell you take. First one is whatever the cell you take. That is, it may be bacteria, protozoa, including virus you can take, no problem, fungi, whatever, irrespective. Cell you take, that cell must contain some of the constituents. Yes or no? So the word is constituents refers to contents. What are different contents? I mean to say that it may be organic or it may be inorganic. Yes or no? So organic or inorganic elements, it may be elements, it may be what? Molecules, or it may be compounds. Yes or no? So you can take any cell, that cell must contain some of the constituents. Those are nothing but organic or inorganic elements, molecules or compounds. See, and I told you people, these all constituents must have their standard level in their body. Standard level. Say for example, some are moles per cell. Sometimes moles per cell. Say for example, not accurately. Say for example, when you take sodium Na+, so that sodium must be 10 gram per cell. Imagine, it's not accurate, it's not correct one. So sodium level 10 gram per cell. It is what? Standard per cell. Sometimes moles per liter. Sometime moles per liter. So Na+, that is 10 gram per liter of blood. 10 liter of blood. This is one of the other standard. Yes or no? So this is what? See, whatever you take the constituents or the contents from our body, they must have their standard level in terms of cell or in terms of blood or in terms of water. Yes or no? Yes. This is one concept. Next, see, the standard must be moles per cell or moles per liter. This is what? Next, important that when you understand the word metabolism, here first we need to understand the word turnover. Turnover. See, here the word turnover means, before that we need to understand these all constituents come under, yes, these all elements, that is organic, inorganic elements, molecules or compounds, whatever, these all come under biomolecules. Yes or no? These all come under biomolecules. So when you take biomolecules, it may be microbiomolecule or it may be macrobiomolecules. It may be macro or it may be micro. So at the same time, these molecules or biomolecules are in the form of metabolites. Metabolites, the first two. So some of the materials are going to form during the metabolism are called as metabolites. Already we discussed in the plants, primary metabolites and secondary metabolites. So the metabolites means some of the molecules are formed by the animals or the plants or the fungus or bacteria during their metabolism are considered as metabolites. Yes. At the same time, now tell me what is turnover. So before we'll entering into the actual definition, we'll discuss some of the points. See, example is general example. Here we have a A. Here the word A. In my example, the word A refers to, it is a complex metabolite. It is a type of metabolite. So this metabolite is converted into B. Means A is nothing but first metabolite and B is nothing but second metabolite. Again it is a metabolite. Yes, second, it is also considered as one of the metabolite. Later B is also being converted into C. Also nothing but it is a third metabolite. Later C become D become finally Z. Z is also considered as one of the metabolite. Simple? Yes. This is called as turnover. Now tell me what is turnover? The metabolites are constantly being changed. These metabolites are constantly being changed into some other biomolecules. Yes or no? Constantly being changed. That is, A is changing into B, B changing into C, C to D, D to so on. Yes or no? So constantly being changed into some other biomolecules. You can take, instead of metabolites, you can take biomolecules. Yes, all considered as biomolecules. Imagine. So it is also one of the biomolecule. Yes, constantly being changed into some other biomolecules, and also made from some other biomolecules, constantly being changed into biomolecules and also made from biomolecules.